Good afternoon to everyone. My name is Keith Golding. Welcome and thanks for joining this webinar today. I'm the chair and Ben Sykes, our executive officer, will be coordinating the webinar. Our speaker today is Professor John Storkey. John is a plant ecologist with a PhD in weed ecology from Nottingham University, and he's a visiting professor at the University of Hertfordshire. He's been working at Rothamsted for almost 30 years, where as well as contributing to the management of the long-term experiments, he leads the agroecology work at Rothamsted that seeks to apply ecological principles to modern farming systems to balance food production with a healthy environment. So welcome, John. It's great to have you on board. For all those joining, can I ask please that you put any questions on the chat facility, which should be at the bottom right-hand corner of your screen. Ben Sykes and I will keep an eye on that and we'll coordinate the questions at the end. And please turn your videos and microphones off because that helps with bandwidth and avoids any problems with losing contact with John. So thanks again, John, for speaking to us. We look forward to what you have to say. There are a good number of people watching and listening. And after, what, 35, 40 minutes or whatever, we'll have some questions which I hope you'll be happy to answer. So thanks to everyone and over to you.
Thank you very much, Keith, and afternoon, everybody. So thanks very much for the opportunity to speak this afternoon. Just looking along the delegate list here this afternoon, I realised that a number of you are familiar with Rothamsted and may be very familiar with the parkgrass experiment, but I've got a few slides at the start to introduce the background to Rothamsted itself and then some of the design principles and history of the experiment before going on to talk about some of the newer science that has been done over recent years. Rather than give you a greatest hits of papers from the experiment, I've very much tried to focus on very recent science, new questions that the experiment has been used to address, but I'm presenting those questions in the context of the very solid foundation of evidence that we've built up over time. So the parkgrass experiment, 168 years old this year, started in 1856. And one of a number of what we call the classical experiments, which were set up at Rothamsted Research here at Harpenden in Hertfordshire in the UK. So John Bennett Laws, the gentleman on your left here, was our founder. John Bennett Laws was a Victorian scientist, a contemporary of Charles Darwin, and also an entrepreneur. And he discovered that by adding sulfuric acid to animal bones, it released minerals and nutrients which were useful to crops, and by adding them to his fields, he could enhance the yields of those crops. And so together with his colleague, Sir Joseph Henry Gilbert, between 1843 and 1856, they set up a series of experiments on different crops, spring barley, winter wheat, hay, all with a similar design, comparing their new inorganic mineral fertilizers to farmyard manure. And a number of those experiments are continuing today, of which parkgrass is one, and we call them the classical experiments. On the left here, you can see John Law's ancestral home, Rothamsted Manor, which is a historic building dating back to the 13th century. If you want to know more of the background of parkgrass and the other classical experiments, you can find them all at this URL in a PDF of a booklet that you can download, and it gives you all of the history and all of the background and the changing plans and designs of the experiments, of the classical experiments, and also of more recent long-term experiments which were set up in the 1960s. So everything that I'm going to say today you should find in this booklet and with additional information as well. So as well as the sites themselves, we also have a fantastic resource of the Rothamsted Sample Archive. So John Laws had a tremendous foresight and wisdom when he started his experiments in the 1800s, in that he kept samples of grain and fertilizer, straw and soil from all of the plots. And we now have those as a legacy and a resource. And they represent a time series going back to those early days of the experiment, right up to the present day, which we can use to analyse and ask additional questions as the technology advances. And some of the examples of the work that I'm going to show this afternoon came from analyses of these archive samples. This is a resource that is available to the research community, and we have a protocol and a system where requests can be made to access these samples and access these archives to challenge new hypotheses and new questions. So we have that physical archive of the samples from all the experiments, including park grass going back to the 1800s. And we also have a curated data archive called the Electronic Rothamsted Archive, which is also open access, and you can download lots of information from the URL that's on this slide as well. So the combination of that electronic archive, where you can go back and you can interrogate data back in time in the experiment, along with parallel data sets like atmospheric deposition of nitrogen and sulfur, meteorological records, the physical samples, and the fact that the plots themselves are still there and we can go out and take new samples and new observations. The combination of those three things is a unique resource globally and has led to a lot of the exciting science that I'm going to be sharing with you this afternoon. This is the most recent review of the science that's been done across the experiments. There's a more recent paper written by Paul Polton on the Broad Bulk experiment that's just been published or about to be published this year. But this paper in Advances in Ecological Research reviews across the classical experiments recent insights and highlights from the literature that were published over the past decade or so, although this paper's a bit old now, from 2016, and we're probably due a new review and a new synthesis of what's been discovered since then. Okay then, so that's the background to Rothamsted and the classical experiments as a whole. Let's focus in a bit more on the park grass experiment. So there's an aerial shot of it. It's on a 3-hectare field at Rothamsted, very close to the manor itself. It was the last of the suite of experiments that Laws and Gilbert set up to test the effect of their mineral fertilizers on yield, comparing it to organic manures, to farmyard manure, set up in 1856. So it's managed for hay. The Rothamsted farm in John Laws's time, about a third of the area would have been grown for grass and for hay to feed the horses, who were providing the labour for the farm. Would have been cut for hay in June, and then the regrowth grazed by sheep. That only continued for the first decade or so of the experiment, after which a second cut was taken later in the autumn, early winter, and that's how it's managed today. 2 cuts, one in June and one in October. So the basic treatment structure is different combinations and amounts of mainly the macronutrients, NPK, with some variation in the micronutrients as well. And an important note is that the nitrogen on the experiment is applied in 2 forms, either as sodium nitrate or as ammonium sulfate. And on the plots where ammonium sulfate is applied, it has the effect of acidifying the soil, lowering the pH on the soil. So very early on, Laws and Gilbert realised this and realised it was confounding the effect, the fertilising effect of the nutrients. And so in 1903, you can see the experiment was split in 2. Longitudinally down the aerial photo we're looking at here. So on the right, I'm going to try and find my laser pointer. Is that working? So on the right there, you can see this half of the experiment was limed, so had chalk applied, and then this half of the experiment was unlimed. So if you look at the electronic archive, you will see up to from 1903, plots were either designated U or L, limed or unlimed. So for example, we have plot 3 here, I think, and that would be either 3U unlimed or 3L limed. And then in 1965, we got a bit cleverer and we split it into the existing 4 subplots, which are A, B, C, D, and they have different target pHs, 7, 6, 5, and unlimed. So that's the structure that we see today. And we know that it's now the longest running continuous ecological experiment in the world. The grass species, plant species that you see on the plots, they haven't been sown, but they are the naturally occurring, the naturally assembled plant community for the soil and the climate in this location, for the local species pool, classified as dicot-rich Cynosurus centaurea grassland. And the fertilizers have acted on filters. So they are selecting out species that are poorly adapted to those treatments. And so the species communities are emerging as a product of subtraction, the filtering effect of the fertilizers. So that's the important point to note and appreciate. So just a couple of shots of historical plans of those three phases, representing those three phases of the experiment before 1903, when the plots were all treated equally with their fertilizers. Then in 1903 split into two, lined and unlined. And then apologies for the blurriness of this particular figure, but from 1960s onwards we have these four subplots. No replication. So this was set up before Ronald Fisher invented ANOVA and factorial analysis and replicated blocks and randomization. So it's a challenge for analyzing the experiment, and we rely very much on regression analyses and time series. So pretty much every one of these subplots is a unique combination of pH and nutrients and fertilizers, although there are a couple of replicants on the experiment. Just to point out the most recent changes in the experiment, these blue arrows indicate what we now call the transition plots. So in the 1990s, the treatments were changed, but instead of losing a whole plot, a plot was split. So plot 14, plot 9 and plot 13 was split. The original treatment carried on one half. On the other half, the treatment changed. So in the case of plot 14 and 9, we withheld nitrogen fertilizer that had been applied continuously up to that point. And a couple of more recent changes that you might not be as familiar with. In 2013, plot 15, which never used to receive any nitrogen, we started applying nitrogen as sodium nitrate at a high rate. So that meant that we then had a series of low, medium and high nitrogen application for both of ammonium sulfate and sodium nitrate. And plot 7 was split, and we started withholding phosphorus because we realized that the phosphorus levels had built up to very high levels on a lot of the plots, and we wanted to ask what would happen if we stopped putting the phosphorus on. So what we're looking at now is the design and the current treatment structure that you would see today if you went out to the experiment. So what that has resulted in is two gradients. We have one, let me find my pointer again. We have one running this way, which are the different fertilizer treatments. So you can see the different shades of green, and also, you know, they've got a white coloration here, which is probably full of hogweed. And so you've selected for the different plant species through the fertilizers. And this way we have lime. So we have a gradient of pH because of the fertilizer applications this way. And the work I'm going to present and the story I'm going to tell over the next few minutes really builds on these three questions. How do these gradients filter biological communities? So how do biological communities, that's plants, soil microbes, pollinators, how are they responding to those two gradients in space? How are they responding through time? So can we see a signal of environmental change? And is there an interaction with treatments? So do the fertilizer treatments, does the fertility gradient and the pH gradient determine the response to environmental change or the resilience to environmental change? And can that be related to the biological communities? And then finally, what are the implications for ecosystem function? Primarily productivity, but other ecosystem services such as regulating services like pollination. So those are kind of the questions we're heading towards. And as I say, rather than do a review of all the papers that have been published over the past 100 years. I've very much focused on very recent papers over the last 3 or 4 years that address those very contemporary and relevant questions. There will be an opportunity, and I'll show this in a slide at the end of my talk, to visit Parkgrass in May, which is an excellent time to come and visit the experiment because a lot of the species will be flowering. But this is just, for those of you who won't be able to make that, this is just a selection of some of the species that we find on the experiment. The overall species pool is in the order of 70, with 40 to 50 of those being flowering forbs. So, for example, we have 4 or 5 species of ranunculus buttercups, that's meadow buttercup on the bottom right. The fritillaries should be just about flowering now. They're one of the first species to start flowering, bugle and germander speedwell. So on some of the plots, the control plot, for example, which is the most species rich, you might find up to 40 flowering plant species. So well worth a visit if you can arrange that. Okay, so on to some of the science. And I've started with, as I say, I'm not doing a greatest hits, but I've started with a classic, because this paper very much establishes the context for everything that we're looking at over the next few minutes. And that is the trade-off between productivity and species richness. So as we move along those gradients, we're moving along that trade-off between selecting for very competitive, productive, fast-growing species and a more stress-tolerant, slower-growing, species-rich sward. So this paper that was written by Mick Crawley and colleagues 15, 20 years ago now was based on the most comprehensive vegetation survey on the experiment, sampling all plots destructively from 6 50 by 25 centimetre quadrats and separating in the lab down to species and calculating relative biomass after drying, combined with regular plot walks to record all the species present on every plot. So it's the most comprehensive data set we have of the impact of the treatments, or those gradients, on species diversity and the trade-off with biomass and productivity. And we repeated the protocol for most of the plots in 2010. Sorry, there's a typo there, and 2013. The methods for doing that and for doing vegetation surveys have remained largely unchanged for 100 years, but I remain optimistic that in my lifetime we may be using eDNA or machine learning or AI to make this a lot quicker. So although at the moment this is still very time consuming and laborious, I'm anticipating that capturing these kinds of data will be a lot quicker in the future. So this is the basic pattern that you see on the experiment, the basic response to those two gradients. You see two processes operating on community assembly and species richness. The first is a biotic stress of competitive exclusion. So as you move along the fertilizer gradient and you add more nutrients, you select for nitrophilous, fast-growing, tall, competitive species, which outcompete the more shade-tolerant, stress-tolerant species. And so you have a reduced species richness through competitive exclusion. And where you apply the ammonium sulfate and the soil pH goes down to below 4, you start getting an abiotic stress, which filters out species that aren't able to cope with the aluminium stress and the aluminium toxicity. So you have those two processes operating on community assembly and operating on species diversity that explains the patterns of species richness that you see on the experiment, and explains that these patterns that you see if you look at species richness across time. I think the striking thing for me for this graph is how quickly those fertilizer effects were manifest. So if you look at the impact of fertilizers, that's the orange and the blue line, you see a dramatic decrease in species richness within the first 4 or 5 years of the experiment, and then those are just compounded through time. An interesting feature of this figure is we're looking at the control plot here, which is the green line, no fertilizer. The dotted line here is where you have chalk added. And there's a decline in species richness between the 1940s and 1980s on the control plot, which is associated with nitrogen and sulfur deposition and a slight acidification of the soil. So we're starting to see the same negative impacts of fertilization, but through atmospheric chemistry and atmospheric deposition. And as I'll mention towards the end of the talk, we are seeing some recovery in that over recent years. But the basic pattern is adding fertilizers reduces species richness through competitive exclusion. Where you have acidification, that also further filters out some species, and that is reflected in a trade-off with productivity. So the colours are slightly different here, but we still have the green line, which is the control plot, which had the highest species richness but has the lowest biomass production, has no fertilizers. Where we add fertilizers, we have this increase in biomass in productivity, but a decrease in species richness. So that's the background, that's the context against which we need to put everything else that I'm going to talk about. And just to finish up on this, a recent paper that I did with Andy MacDonald started with that trade-off between biomass and species richness. We've taken out the acid plots here, which is why we get a stronger regression. And we also make the point in this paper that that cascades down to soil carbon as well. So because soil carbon is related to the rate of photosynthesis and the amount of carbon going into the system, there's a relationship between carbon sequestration or carbon capture, which is a very hot topic at the moment. How much carbon can we sequester in grasslands? And the trade-off is you actually have to have high productivity, so you might have to put some nitrogen on, which is this bottom right-hand panel, to get that increased carbon sequestration. So we've got these inherent, these ingrained trade-offs along these gradients on the experiment. And one of the things that we're interested in is where are the residuals, where are the outliers, where are the plots that might break or solve some of these trade-offs. And in the paper we use plant traits to explore those, and I think I might return to it a bit later on. Okay then, so there have been big shifts in species richness in response to the fertilizers with a trade-off with productivity. We also know there have been dramatic functional shifts and also shifts in functional diversity. And this has been a topic of some of the more recent work that's gone on. This was an analysis that I included in the Advances in Ecological Research paper that just makes the point that you can interpret those gradients on the experiment, both on the park grass experiment and the broadbulk experiment, in the context of Phil Grimes' plant ecological strategy scheme. If you know about that, he basically said there are three plant ecological strategies. You can either be a competitor, so you're adapted to fertile, undisturbed environments. You can be a stress tolerator, you're adapted to infertile, undisturbed environments, or you can be a ruderal, adapted to disturbed, fertile environments. So those two gradients of disturbance and fertility, when put together, can explain the functional composition of any plant community. And so without worrying too much about the detail of these ordination plots, so each of the points on the plot is an individual plot, either on park grass, which are the open circles, or broadbulk, which are the closed circles, which is an annual system. Park grass is a perennial system. And for each of those plots, we've calculated the community weighted mean, or a composite measure of lots of different plant traits which are indicative of their strategy. And so if you look at park grass compared to broadbulk, what it does is it discriminates between your annual ruderal plants, which are adapted to a short life cycle, fast growth, high nitrogen acquisition, from the longer lived perennial species. So that's a gradient of disturbance between broadbulk and park grass. But of more interest today is if you then just focus in on broadbulk, which is the plot on the right, what you end up with is that gradient of fertility. So if you look across all the plots, you can discriminate between the plant communities based on their traits, And what you see is a selection on plots that receive lots of nitrogen and lots of phosphorus, or farmyard manure, selects for traits that are adapted to rapid growth and resource acquisition. So they're tall, they have a high specific leaf area, and plants that are adapted for resource conservation. So they grow more slowly, but they invest more in leaf defenses. So they're less palatable, longer lived leaves, lower specific leaf area. So we see that gradient, but an important point to make is these are the species that are dominating here. These are the species that are dominating here. But of course, on the low fertility plots, we have all the plant ecological strategies represented, but in a more balanced way. As we add the fertilizers, we are systematically removing some functional types from the community. So we're leaving functional gaps, which is why the residual plant community is dominated by the traits that are best adapted to that particular plot and that particular environment. Okay. So what's the consequence of those functional shifts, or the consequence of that reduced functional diversity, or those functional gaps for higher trophic groups? Now, most of the work that I'm talking about, I've got a reference and I've got a DOI at the bottom that you can look this up. The next couple of slides are actually from a PhD thesis. They might be a bit harder to track down the source material, but if you get in touch with me, I can send you a copy of the thesis. But this work was done by Thomas David, who was a PhD thesis with myself and Carly Stevens at Lancaster, looking at the consequences of that filtering of the functional traits of the plants by those gradients of fertility and pH on the pollinator community. And interestingly, there's another paper from Sussex University that should be published very soon that adds to this work and builds on it and identifies some of the species specific responses. But the backdrop to this was, let's start with those plant response traits. Let's start with that functional shifts and functional filtering in response to eutrophication and acidification, and ask what are the implications on the traits that are important for pollinators, the plant effect traits, nectar production, flower symmetry and morphology, flower color. So I realize you probably can't digest all the information in this table, but it's illustrative of the fact that here we have all of the, sorry, let me get my pointer again. Here we have all of those functional effect traits. And so what Tom did was he built a model that looked at the overlap between the effect of the fertilizers on the response traits and the overlap with the effect traits, to look at what the impact, for example, of adding the nitrogen in different forms was on these effect traits. And if we just focus on the sodium nitrate for now, because we don't have the confounding effect of pH, what was apparent was that there's an empty circle here around nectar production and nectar sugars. So this was looking at a low rate of nitrogen at 48 kilograms of nitrogen per hectare. It wasn't actually affecting the overall production of nectar and nectar sugars, but it was affecting the quality of that resource, or the diversity of that resource. So because it was acting as a filter on the species community and it was selecting out some species, you had a reduced diversity of flower forms, you had a shift in the flowering color, and you were losing those long corolla flowers, which are particularly important for long-tongue bees. So eutrophication and acidification was filtering out, was systematically removing some functional types of flowers and leaving functional gaps. And I think that's the key point that was coming out of this work. He also looked at flowering phenology and found effects of the fertilizers on the timing of those flowering resources, both interspecific differences, so filtering out perhaps the early flowering species, but also intraspecifically. So the same species, for example, black knapweed, was flowering earlier on some plots and later on other plots, and that was an effect of the fertilizers. So we have a really nice data set actually over, I think, 12 years now of the flowering periodicity of all of the species across all of the plots. And you can derive these kinds of graphs which show that the application of nitrogen in the different forms affects the availability of the resources in terms of how early they're available, but also the number of different flowers that are available and the number of species that are flowering at any one time. Here he's overlaid that with the demand from bee activity and the demand for those resources. And again, this is kind of a summary figure, but in the thesis, in the chapter, he decomposes that to look at what are the specific effects of pH, the specific effects of increasing nitrogen rate on the availability of those resources. But the net effect of all of that, and I think this is one of the key points that I want to get across this afternoon, is the effect of the fertilizers is reducing functional diversity and it's leaving these functional gaps. So we get a functionally simpler community which is dominated by certain traits, and that's reflected in the communities of pleiotropic groups that are using those plants as a resource. So here we have a network analysis. So we have the plants on the bottom in green. You probably can't read the species names, and then the pollinators that are using them in blue for the control plot on the left, and for the plot that receives the low rate of nitrogen. And you can see how we've got fewer, the diversity of the resources is reduced, is simpler, so the networks are simpler. And I think an open question is so what does that mean for the delivery of ecosystem services of pollination by that pollinator community, or the resilience of that community? And we see a very similar story for soil microbial diversity. So here's a paper from 3 or 4 years ago. Not my area, so forgive me if I gloss over this a bit. But this work was based on new data from concurrent sampling of 2 plots, a fertilized plot and an unfertilized plot, in a nested sampling scheme at multiple scales. But the key idea was to associate soil microbial communities with plant communities. So we did parallel sampling of the soils and sequenced the DNA of the soil microbiome, and at the same time we did a more conventional vegetation survey at these different scales. And the authors did some clever network analysis statistics to associate, first of all, associations within the communities, the microbes and the plants, and then between them. And they found a very similar story to the pollinators, in that if you compare the unfertilized to the fertilized plots, you just get a much simpler network. You create these functional gaps. So what we're looking at here are associations between different functional groups of the soil microbiome. So, you know, I confess that these words mean something to me, but I don't know fully how the specifics. But the general point is that where you've got lots of grey circles, that means that you've got a significant positive or negative correlation, I can't remember what, and then the orange is the other. But you've got lots of significant associations. Most of the circles are filled in. Whereas when you add fertilizers, it's as if you create all of these blind spots in the network, and you're systematically deleting parts of that network, and you're left with all of these empty gaps. And again, I think that raises the question of, so what? What does that mean for ecosystem function? What does that mean for resilience? And if you look through the paper, you will actually see there are some figures that analyze the abundance of functional genes. So the authors also argue that fertilizers, as well as affecting the network, have also affected the functioning of that network in terms of the expression of some of the genes that are associated with nutrient cycling. And they also associate these microbial communities with plant species, where they can find statistical associations and relationships. And again, just makes the same point. Look at the number of connections on the left. We've got more plant species that are significantly associated with different microbial groups. And on the right, we've just got Arenatherum with a much simpler network. So same story. The fertilizers are systematically removing some functional types and creating these functional gaps in biological communities and biological networks. So what? Is that leading to consequences for the resilience, for the functioning of those swards, for those different plots? And so the last few examples of recent work I've got are looking at that, are looking at yield responses, are looking at the consequences of those shifts in biological communities for functioning. So this is a really nice paper. It's a statistical paper that was written by one of our statisticians, John Addy, here at Rothamsted. Don't ask me what heteroscedasticity means. I think it's something to do with the fact that variance or standard deviation isn't constant across the data set. So it's a way of dealing with that non-parametric data. But so the paper develops this really nice novel statistical approach to look at yield variance in time on the parkgrass experiment, relate it to variance in weather, so yearly temperature, yearly rainfall. But the novelty is John looked at slicing those data in different ways to find the best fit to the model and the best predictive power, and then using climate change scenarios was able to forecast what future yields might look like. And you might begin with the hypothesis that climate change with increased carbon dioxide concentration and with increased temperatures might lead to increased yields. You might get higher productivity. But actually what we're observing on the experiment is a consistent but steady decrease in yields. And this seems to be consistent across most of the plots, and John was able to model it and associate it with weather and the frequency of warmer, drier years. The big driver of yield variation on parkgrass is water stress, moisture stress in the early summer. So if we get hotter, drier springs and summers, we see lower yields. So in the paper, John doesn't really ask the question, what is the differential effect of the fertilizers on this prediction? Although you can see, okay, you say, oh, the slope in the nil plot is probably lower than in the fertilized plot. Obviously, the yields are lower as well. I think there's some additional questions to ask about whether the more diverse plots where we've retained more functional diversity means that they are less vulnerable to some of these impacts of climate change. And not the next paper, the paper after that addresses that and provides some evidence that that is actually the case. Okay, so that's an empirical statistical analysis of the fact that yields have been going down, and it may well be that some plots are less susceptible to those impacts of climate change than others because of the retained functional diversity. This is a really nice paper that dug down into the potential mechanisms, plant physiological mechanisms that might explain that reduction in yield. So this was an analysis of those archive samples, and through some clever isotope analysis, they were able to derive stomatal conductance or differences in stomatal conductances through time. So they looked at two periods, 1917 to 1931 and 2004 2018, so a historic and a contemporary period, and they compared stomatal resistance, stomatal conductance. So that's what this is here. It's the internal concentration of CO2 compared with the ambient concentration of CO2. And they classified the plots as either dicotyledon rich or grass rich. So that's what they've done in this plot over here as well. So as we've seen already, the fertilized plots, we basically have the fertility gradient here. The fertilized plots are dominated by grasses, and the forbs are found on the plots with lower fertility. What they found was across the board, so if we look at this, let me get my pointer up again. Across the board, and if we look at this top panel here, water use efficiency increases or improves across the experiment. So I'm not going to worry too much about the units here, but this top set of panels is water use efficiency. So water use efficiency improves across the experiment because CO2 concentration has gone up, which means that you've got more opportunity to control your stomata, to control water loss while maintaining your concentrations of CO2 in the internal cellular pores. But what they found was that yield has gone down between these two periods, and that was what John Addy had found as well. But they found that yield had gone down more, and it was actually only significant between these two periods on the grass-dominated plots. And it was the grass-dominated plots where they also observed this historic reduction in stomatal conductance. So it appears that the grasses were more sensitive or responsive to that increase in ambient CO2. That had led to reduced stomatal conductance, which then cascaded down into reduced yields on the grass-dominated plots. So again, it's providing some evidence that these plots down here with lower soil fertility, with greater functional diversity, were actually holding on to their yield better in response to this environmental change, this increased carbon dioxide concentration in the atmosphere. And this bottom panel here is just nitrogen acquisition or nitrogen use efficiency. So the other point they make in the paper is one consequence of this reduced yield is less of the nitrogen fertilizer is finding itself into biomass or into the hay or into the silage, which means that you've got more losses to the environment. So there's a nice story emerging here. I think there's obviously lots of questions remaining, but you've got the impact of fertilizers on the functional shifts in the plant communities leading to these functional gaps. That's having consequences for the differential physiology of those functional types, which is cascading down to impacts on yield. And I think I'm just going to show one more, and then I'll probably draw things to a close because time is getting on. Again, a very recent paper. So we've looked at the trends in yield over time. We've looked at how that's related to differences in the physiology between the functional types which have been selected for by the fertilizers. This particular paper was looking at variance in yield to short-term perturbations in weather. So those long-term trends we were seeing were consequences of increasing CO2 over decadal timescales or increasing temperature or changes in rainfall over decadal timescales. Here we're looking at what about those year-to-year perturbations and stochasticity in weather. Are the different plots more or less resilient to them? And Jonathan Silvertown did some really nice work on this in the 1990s. This is really building on that and using a slightly different statistical technique to ask the same question. And in the paper, what they come up with are figures like this. And the key point about the measure that they used was they were able to separate variance from absolute yield. So although a plot might yield lower and so will inherently have a lower coefficient of variation, they were able to separate that such that you can just compare the plots on the basis of their temporal yield variance. So let me get my pointer up again. So the bars here are variation in yield, and the fitted line and the crosses here are that mean yield. So these are for all the plots. So again, you see the same trend that John Addy found. We get this steady, gradual decrease in yields. And then on the bars are showing how variable those yields were within, I think these are 5 yearly slices of data, and they've looked at how variable the data are within those. So the striking thing that was, apologies for my phone, hopefully that will stop in a bit. So the striking thing about these data were that we had this unimodal distribution where we have high variation in the 1990s and then low either side of that. We really didn't expect to see that unimodal pattern. So the question is, that's for all the plots. The question is, are there differences then between the plant communities and between the individual fertilizer treatments? And indeed there were. So what we're looking at here is a plot that receives PK NAMG but no nitrogen, so it's dominated by legumes. It's got a high legume component, more diverse, and down here a less diverse, more fertile plot. In this case, it's farming up in Europe. But if you look at the paper, you'll see clear differences between the plots in terms of their absolute variance over time and how clearly you see this unimodal pattern. So there's something striking happening here where you've got these legume dominated plots. You have low variance in recent years, but you have high variance on the more fertile plots in more recent times. So what the paper explores is the contribution of biodiversity to that. And so we did find that the more biodiverse plots were less variable and so were more resilient to that short-term perturbation in weather. And this unimodal effect was explained by a period of rapid environmental change over that period. So this is a graph of, let me get my pointer again. This is a graph of temperature recorded at Rothamsted, at the Met site at Rothamsted, and you can see the period that we're looking at, where we had that peak in yield variance, was where we had this rapid increase in temperature over the 1980s and 1990s. And we also saw these marked decreases in nitrogen deposition over that same time. So we had a period of environmental change, which coincided with that yield variance. And in the paper, you can look at the height of that peak and how resilient different plots were to that short-term perturbation or that change. And now I think the really interesting question is how adaptable those plots are to long-term shifts in climate and long-term shifts in temperature and rainfall patterns. I'm very conscious of the time, so I'm going to just skip through those last few slides. We might have an opportunity to return to them later. But I wanted to hopefully pull things together, and my intention was there was a common thread running through those studies, those recent studies, of the impact of the fertilizers on environmental gradients, on biological communities, and on ecosystem function, specifically resilience to short-term perturbation and adaptation to climate change. Because my closing slide is to make the point that against that backdrop of the challenges that we're currently facing, that the parkgrass experiment has never been more relevant and more useful to addressing these questions. And I've just come up with these 3 questions of my own to close. How do we sustainably integrate ecosystem services into improved grassland, given that we've created these functional gaps through intensification? How do we knit back that function into our agroecosystems? What are the implications of those functional gaps, of that loss of functional diversity for ecosystem function and services? And what is the contribution of biodiversity or functional diversity to the resilience of grasslands to environmental stress and the capacity for adaptation? So apologies, there was a lot there. We covered a lot of material, but as I say, all with DOIs. So if any particular study interested you, you can go and look at it in detail. But even though perhaps you weren't able to grasp all the detail of each individual study, hopefully it will have served as an illustration of how important experiments like parkgrass are, with that continuity of data and that continuity of treatments to elucidate these long-term responses to environmental change. And I'll close with this slide, which is advertising the opportunity for a visit to Rothamsted, where you can come and see the archive and you can come and see parkgrass in May when it's in full bloom. So I would encourage everybody to make a note of that and to book if possible. And with that, I'll stop sharing, Keith. Thank you.